Barium is a contrast medium, which the patient takes orally or as an enema to enhance visualization of the gastrointestinal tract on a radiograph. In this radiograph, the large intestine is clearly seen. Teeth grind food during mastication. The exposed part of a tooth, or the crown, is covered with enamel, which is the hardest substance in the human body. The crown consists of rounded projections, or cusps, that facilitate the grinding process. Over time, food particles can cling and accumulate in the grooves between the cusps and between the teeth, forming a sticky plaque. Bacteria, such as Streptococcus, will flourish within the plaque, eating and digesting the sugars and carbohydrates. As a result, bacteria produce acidic byproducts that are harmful to the teeth. Normally, saliva will neutralize these acids, but plaque provides a barrier that prevents saliva from reaching acid that is concentrated around the tooth's enamel surface. From a cross-sectional view, the tooth consists of the exposed crown supported on a neck with roots that project into the mandible or maxillae. There are three tissue layers that make up the tooth: the hard enamel, the dentin, and a central region known as the pulp. Bacterial acid from plaque begins to erode the surface of the tooth, dissolving the enamel and forming a cavity or dental caries. This begins the process of tooth decay. The enamel contains no cells, so no pain is experienced at this point. As the cavity progresses, bacteria can penetrate the enamel and dissolve a crater in the dentin layer. Eventually, the cavity is extended into the soft pulp, where the destruction of pulp tissues leads to inflammation and infection. As extensive destruction continues, a pulp abscess made up of pus and dead tissue may form. Stimulation of the nerves causes the sensation of toothache and eventually severe pain. The abscess can spread into the surrounding bone and could cause serious problems if left untreated. Endoscopy is a procedure used by physicians to view, sample, or repair the passageways of the digestive tract. In this video, the endoscope is introduced through the oral cavity, passed through the pharynx and into the esophagus. We are now passing through the gastroesophageal junction. Notice the rugae lining the stomach. Bile is stored in the gallbladder. Normally, the cholesterol in the bile remains in solution. Under certain conditions, the cholesterol may precipitate and form solid crystals. If this process continues, the crystals grow larger and become gallstones. The stones may block the flow of bile, causing pain and jaundice. Gallstones may be removed surgically. Normally, gastric juices are secreted in the presence of food. Stress and smoking can also cause the stomach lining to secrete gastric juices. In the absence of food, the acidic juices erode the lining of the stomach, leaving an open sore that may bleed. This open sore is called an ulcer. Recent research has shown, however, that a bacterium, Helicobacter pylori, may be a major factor in ulcer formation. The use of antibiotics to kill the bacteria has proved quite helpful in treating many patients with ulcers. Digestion begins in the oral cavity with both mechanical and chemical breakdown of food. Chewing motion breaks down food particles and mixes food with saliva secreted from salivary glands. There are 3 salivary glands that contribute secretions to saliva. The parotid glands contribute a serous fluid rich in amylase. Amylase is a digestive enzyme that begins carbohydrate digestion. The submandibular glands secrete a primarily serous fluid that also contains some mucus. Mucus secreted from the sublingual glands helps bind food together and acts as a lubricant during swallowing. As food is swallowed, it enters the esophagus and is moved downward by a wave-like muscular contraction called peristalsis. The contractions progress from the superior end of the esophagus to the gastroesophageal junction, where the contents of the esophagus empty into the cardiac region of the stomach. The lining of the stomach secretes gastric juices that contain digestive enzymes. Peristalsis churns and mixes the food with gastric juices while moving the food toward the first part of the small intestine, the duodenum. In the duodenum, food is digested even further. The bile duct from the liver and gallbladder and the pancreatic duct from the pancreas all empty their secretions in the duodenum. The bile works to break down fats, while the pancreatic enzymes aid in the digestion of other food particles. Together, these digestive juices help prepare the food so that the small intestine can absorb as many nutrients from the food as possible. The majority of the absorption takes place in the second portion of the small intestine, a 3-foot section called the jejunum. As the food travels through the jejunum, it's gradually broken down into smaller and smaller particles. Folds line the walls of the intestine and act to increase the absorptive surface area. The green nutrient particles and the yellow fat particles get caught in these folds and are absorbed into the intestinal wall. Larger, less digested particles are pushed onto the large intestine, where much of the water they contain is reabsorbed before being expelled from the body. A closer inspection of the folds reveals that the intestinal wall is covered with thousands of finger-like villi. A network of tiny blood vessels called capillaries runs just below the surface of each villus. These capillaries absorb the available nutrients in the intestine and transport them to the liver. The yellow fat nutrients are absorbed into a vessel called a lacteal that carries them to the circulatory system. Nutrients absorbed by the blood in the villi are carried to the liver by way of the portal vein. In the liver, the blood is filtered and the nutrients are extracted and processed. After processing, the nutrients are either stored for future use or released into the bloodstream to be used throughout the body. Upon closer examination of the stomach, the muscular folds called rugae that make up the stomach lining are clearly visible. The rugae gradually smooth out as the stomach fills, permitting stomach distension. A cross-section of the stomach lining reveals that in between the rugae are gastric pits, which are the openings of the gastric glands. The gastric glands are lined with different types of cells that contribute various components to gastric juice. Chief cells and parietal cells make up the gastric gland. Pepsinogen is secreted by the chief cells, and the parietal cells secrete hydrochloric acid. Hydrochloric acid converts pepsinogen into pepsin. Pepsin is the key component to gastric juice and is a potent digestive enzyme. By splitting protein molecules into even smaller parts, pepsin begins the digestion of proteins. The lining of the small intestine is characterized by numerous circular folds called plicae circularis. The plicae are lined with finger-like villi. From a cross-sectional view, the villus contains a network of capillaries which surround a specialized lymphatic vessel known as a lacteal. The epithelium of an intestinal villus consists of columnar cells which are covered with microvilli. This succession of folds and projections increases the surface of the intestinal lining for efficient absorption. You may now watch carbohydrate, fat, or protein digestion and absorption as they occur on the surface of an intestinal villus by choosing a button. Carbohydrate digestion is completed by enzymes in the small intestine. Carbohydrates are absorbed by the villi and then enter the capillary. Fat digestion occurs primarily in the small intestine. Fat molecules are digested and absorbed into the epithelial cells of the villus. The fats are formed into clusters called chylomicrons, which pass into the lacteal. Lymph carries chylomicrons away from the villus. Protein digestion is completed in the small intestine. Proteins are broken down first into peptides, then into amino acids. These are absorbed into the villi, then into the capillary. This is a histological image of a section of a villus in the duodenum. This is a histological image of a section of the wall of the duodenum. This is a histological image of a cross-section view of the wall of the esophagus. This is a histological image of a section of the wall of the fundic region of the stomach. This is a histological image of a section from the wall of the gallbladder with an adjacent piece of liver included. This histological image from the liver illustrates many of the components of a hepatic lobule. This is a histological image of a section through the submandibular gland. In this histological image from the tongue, you can see an example of dense, irregular connective tissue. This is a histological image of a section through a developing tooth.